Hello, everyone. I am here representing the team at California State University Stanislaus. Our project was on OPGAL-WASP Open Science. We were training citizen scientists and creating or contributing to an open database. So our principal investigator is Esteban Montenegro. Dr. Montenegro is the faculty director of the Interdisciplinary Statistical Outcomes Lab. I am Jennifer Frisk. I'm the project coordinator on this project. I am also a graduate student in Dr. Montenegro's ISO lab. And then Jose Diaz-Garayua is our faculty consultant on this project. He's been invaluable. He is the director of the Human Environment Exploration Lab, and my husband, August Frisk, who was our field coordinator, created our materials. Wonderful guy, I'm biased, but he is a student in Dr. Garayua's Human Environment Hex Lab, as they call it. So, all right, so I'll tell you a little bit about this project. Our inspiration behind it, my husband and I originally had this idea to create workshops to identify oak galls, which if you don't know what that is, that's part of the reason we were doing this work, is most people don't. We had, over COVID, our three kids and us were trapped in our house and didn't get out much. And just for the sake of mental health and for something to do as a family, we started kind of exploring our campus and walking around. And we're very lucky here at Stan State to have they call it the Trans-California Pathway. So it's kind of a transect of all of the different native plants at different elevations of California. So it's a really cool thing that we have here. And the end of the pathway is a valley oak grove. So there are, I think, about 20 trees, and it's just a little mini oak grove, but they're all valley oaks and they're native to our area. And so during that time when everything was shut down, that still was open to the public. We were able to take our kids there and we learned how to use iNaturalist. We were able to make lots of connections through that weren't just other citizen scientists, but even research labs that were using the tool. So anyway, in interdisciplinary and interinstitutional backgrounds, we are students here at Stanislaus State, but we do have lots of connections with other institutions. We work with the Forbes Lab. We actually met them through iNaturalist, working on galls. It's such a unique niche topic that we were able to work with labs that are on the cutting edge of that research. And so the Forbes Lab is at the University of Iowa. We've worked with them for several years at this point. August works with the Dos Rios. He's a docent at Dos Rios, which is the California's newest state park, which is really cool. They also have valley oaks there, so we get to do a little bit of studying there. And then he also works at River Partners as a restoration scientist. And so this is all just very personal work for us. And we're really excited about the opportunity to bring it to a wider audience. Okay. And the significance of this work. So aside from, again, furthering research on an understudied phenomenon, most people don't know what oak galls are, or that they, are produced or induced by wasps. Or even if you hear the word wasps, you think of something that's going to sting you or like a really scary big thing. These are teeny tiny guys that do not sting. They're, actually really beneficial to, and a really important part of the biodiversity that's found on oak trees and within oak trees. And then on top of that, we all four of us, our entire team is really invested in citizen science and open research for its social justice significance, that, anybody can be a researcher, anyone can be a scientist. It shouldn't be that it's locked behind. You shouldn't have to be affiliated with an institution or have a degree to do some of the work that we do. So, and changing the culture of higher education. So thinking especially in the sense that we come from public universities, the campus is open to the public. Our Trans-California Pathway and the oak grove that we have is open to anyone. It's not behind a fence or anything like that. We are publicly funded. We should be open as well. And so thinking about how can you open access to lab spaces or technology or certain techniques or training that maybe might be reserved for students or faculty or people that are more traditional researchers, how we can make that more equitable. and also produce quality data from that too. Those are two different things, but hopefully we're finding solutions to that. So, all right. So our project goals, what we actually wanted to produce from this project, we wanted to create training materials that would train people on what oak galls are and then also how to identify them. We also wanted to do our citizen science workshops. So a big goal of ours was citizen science is great. It's wonderful using tools like iNaturalist, which are open to anyone. They don't cost anything. You don't have to be affiliated with an institution to sign up for an account. They're it's great, but the quality of data that they produce sometimes is not really entirely usable or that helpful. So thinking about how can you partner citizen science and open tools with actual training and protocols and bring the community in so that they contribute in a more meaningful way and that their data is actually usable. So and then contributing to ongoing research. So you'll see this picture right here. It's a snapshot of what the Forbes lab at the University of Iowa sends us. So we provide them with samples from the field, we ship them, and then they in turn share their data back with us. So they do genetic sequencing on these galls. And they actually, they're producing lots of research from it, lots of publications, all sorts of new. This is a phylogenetic tree that they have just updated. That's a very recent update, that screenshot. And so it's really fun to get to see that. And it's not, again, not reserved for just the people in their lab. They send this out to anybody that's contributed to their data points on iNaturalist, but especially those who mail in samples, specimens. And then this picture down in the corner, we brought some citizen scientists out to the field with us during our training. Okay. And so then thinking about outcomes, if we keep our goals in mind, so these are the goals we had, we were able to create training materials, which if you scan that QR code, you can actually get to a digital guide. We printed physical ones so that we could use them in the field. We left the backs blank so that people could write notes and things like that. People really loved being able to take those home because then when they go out with their family or their friends, they can use it in the field still, which is really fun. But the digital guide is a little more permanent. You'll have access to it forever. And it's a resource that we were able to also then share really easily with other institutions like Dos Rios, the state park. They're currently using that for leading little walks on Saturdays to teach people about oak galls, which was a really, really rewarding outcome from this. So it kind of spread beyond our project, which was really cool to see. And then that was a flyer for our workshop, two of our workshop dates. We actually ended up doing, so those two were open to the public. We advertised them out to everybody. Instagram, our institution actually advertised for us as well, which was really helpful. But we also were able to partner with the Geography and Environmental Resources Department here, and they brought some classes out too. So we ended up having more dates that involved those undergraduate students, which was really fun. And so it was more kind of more like a classroom, like a lecture style that we did for those dates. And then citizen science workshops, we were able to do the workshops, which was wonderful. And the sense of community, that's the biggest thing I think was kind of surprising to us. Our first day that we went out, there were only 5 people that showed up. And so we thought, ooh, it was great. It was intimate. Those 5 people were very interested, learned a lot. But it was kind of, to us, we were hoping for a little bit bigger of a turnout. But then the very next weekend, those people kind of spread the word too. And so from word of mouth and everything, we went from 5, I think, to 23 that next Saturday. So that was really, really cool. And then also the 5 people that came, it was really interesting that our connections to those people were through things like, the river restoration place that August works at, Dos Rios, knowing people that volunteer there or work there. These people that this information isn't going to just die with them, they are going to then spread it to other places where it will continue to grow and spread, which that again has been the most rewarding part of this, surprisingly for me. And then contributing to ongoing research, we did, let's see. Okay, so contributing to ongoing research, Garen Brown, the top student there, that is a graduate student at the Forbes lab. He is the one that makes sure that he communicates back with the citizen scientists. And so he has they have an NSF grant that helps them to do the genetic sequencing and all of the research that they do in their lab. But part of the stipulation of that grant, similar to ours, is that there's a component of it that has to be open to the public. And so what they do for us citizen scientists who have been helping or who have been interested in this work, they do twice a year updates they over Zoom where everybody can come and learn about what's been happening. And so that's been really awesome. Garen has been a wonderful person to partner with. It's also really cool to see that the citizen science data that we're helping to collect and that we're training people to contribute to, that it's actually like producing new knowledge every day, which is really cool. And then the Forbes Lab contributions, this sheet right here, I have a link to it so I can show. We created this, so this was part of our project as well. We wanted to have an open database that cataloged what we have been contributing. So through this project, our ORCA funded project, we have contributed all these collection IDs. So we went out into the field, we made the identifications, we collected samples. The ID number is associated for the iNaturalist ID. And you write it on the sample, and then when you send it into the Forbes lab, they're able to connect those two things. So they get all sorts of information. They get geographic data, they get the time, the seasonality, all sorts of what the host plant was. It's very, very helpful. They can take that research in a lot of different ways. And it was nice for us to be able to create this because it's kind of reinforcing to our workshop participants that their data isn't just, I don't know, sitting there and dying on iNaturalist. It's being used, it's being cataloged, and it is appreciated. So, okay. Yes, so a huge thank you to ORCA. We could not have done this project without ORCA. We might have been able to do maybe like one workshop on a Saturday type of thing. I don't think we would have been able to spend as much time as we did on the workshop materials, the flyers, the taking the time to be really intentional about connecting with our networks if it wasn't funded the way that it was. We were able to pay 3 student researchers to participate, to train people, to collect specimens, to do the shipping. That also, I think, makes the work seem more valuable as well, that it's not just volunteer work, it's actually valuable and worthy of pay. And it's a justice issue as well, that students often are tasked with volunteer work, internships that are unpaid. It's nice to be able to pay that for students that need the funding. So, and then I think also one of the coolest things for me personally about being affiliated with ORCA with this project is that we got to brag about that and talk to people about what ORCA is and what open research is. So all of our workshop participants, we made sure to explain to them how it was funded and that they were a part of this open research community. And that's been really rewarding, and seeing them be so excited about that as well. There's definitely a hunger for and an interest in open research out there in the community. And then I do also want to thank our faculty partners at our CSU. So Dr. Montenegro and Dr. Gariua, the institution is a little challenging to work through. We are a teaching university. We're not a research university. So the grant process is a little tricky, and some of the bureaucracy is a little hard to navigate. And they've been wonderful throughout the whole process. And that's another really wonderful benefit of ORCA, is that their time was also compensated. So I think they would have been invested either way. I love them, but at the same time, that work is valuable and their time is valuable. And I'm really happy that we were able to compensate them for that. So I would encourage anybody with an idea, whether it's a substantive idea, like what we had with the oak galls, wanting to teach people about that. If you have something like that you're interested in, please apply to ORCA. It's wonderful. It's been a wonderful experience for us. Meeting with the teams every so often, a couple times a month, sometimes, that has been really inspiring to see what other projects are doing. And also, yeah, it's just, we are definitely looking forward to applying in the future as well. So thank you very much. This has been a wonderful experience.